Here is the answer key for the DNA and DNA replication review sheet that I have in your overview packet. We completed this after we talked about DNA and when DNA replicates, and then we made a model of DNA out of Connects, and then we replicated our Connects DNA molecule. So this was just a review sheet to kind of make sure that you understood what we were talking about when we replicated our strand of DNA. So the first question is, what does DNA stand for? And it is deoxyribonucleic acid. All cells have DNA, prokaryotic and eukaryotic. Remember, in prokaryotic cells, which are bacteria, they have DNA, but it's not in a nucleus. It's in a smaller capsid that swims around in the cytoplasm. In eukaryotic cells, you're going to have your DNA located in the nucleus. The monomer of DNA is a nucleotide. Remember from chapter 2, the monomer is a nucleotide and the polymer is nucleic acid. So the nucleotide contains the 5-carbon sugar, which is deoxyribose. It contains a phosphate group, and it contains one of 4 nitrogenous bases. Not all 4 at once, just one at a time. It's either adenine, thymine, guanine, or cytosine. The bases of the DNA molecule, like we just said, are adenine, guanine, cytosine, and thymine. Remember, letters that have straight lines, adenine and thymine, they are always paired together according to the rules of base pairing, and nitrogenous bases that have curvy lines, like a G and a C, they are also paired together. To complement this strand of DNA, the A is going to pair with a T, T with an A, T with an A, C with a G, T with an A, C with a G, G with a C, A with a T, T with an A, C with a G, G, C, T, G, G, C, A, T, A, T. All right, if DNA is double-stranded, what connects these 2 strands? And it says right here in this picture, the hydrogen bonds hold those complementary base pairs together. You can see your sugars and phosphates back here, your sugar-phosphate backbone. And you can see your nitrogenous bases on the inside. T's always pair up with A's, C's always pair up with G's. A genome. You may have heard of the Human Genome Project. A genome is a complete set of genetic information for an organism. So it's mapping all of an organism's genetic information. The hierarchy of DNA, you've got your nucleotides, which come together to make up your strand of DNA. In a strand of DNA, you'll have a section which is called a gene. We'll come back to that in a second. 3 DNA subunits at a time make up a codon. So the DNA is going to be copied onto a strand of RNA for a codon, and let's say it's AUG. That codon is going to code for an amino acid, let's say methionine. All of these amino acids are going to be linked together in a chain, and that chain makes a protein. The protein in turn is going to code for a trait. Okay, so nucleotides make up DNA. DNA puts its code on the RNA. RNA is going to piece together the amino acids. In a chain of amino acids is a protein. Proteins code for a trait. DNA replication is when DNA copies itself. We talked a lot about this in chapter 5. DNA copies itself for DNA replication in the nucleus. It occurs during the S phase of the cell cycle. Enzymes are what control DNA replication. DNA polymerase specifically. Okay. And then DNA is called a semi-conservative process because the original is used as a template to make 2 new strands of DNA.